let's take some notes together on enzymes. This covers 3.1 through 3.3. Enzymes are protein molecules that are produced by living organisms. So you've covered enzymes back in 9th grade, and you should remember that enzymes are serving as biological catalysts within our bodies or in all living organisms. And that word catalyst, okay, let's talk about our definition for catalyst real quick. A catalyst is something that speeds up a reaction. Okay, so it's going to be anything that's speeding up a chemical reaction or speeding something up. So enzymes serve as biological catalysts. Now, they are either going to cause or accelerate chemical reactions in living organisms. Okay, so that's one of our key idea we need to understand about enzymes, is that they are either causing or accelerating chemical reactions. The next thing that we need to know about enzymes is going to be enzyme specificity. Alright, so we have a label diagram over here of our enzyme. You know where your active site is, and we should know that our active site is directly associated with our substrates. Okay, what does that mean? It means that your active site is directly linked to the substrate that's going to match it. Okay, so substrates, these are your target molecules. So the targeted molecule that your enzyme is going to work on. And these substrates are converted into products by the enzymes. Okay, so enzyme specificity, the concept comes from the fact that enzymes are highly specific. Okay, so your active site, you see how your active site matches your substrate. So this also brings in the lock and key model. Okay, that's the concept that in enzymes, each enzyme, it will only catalyze that one specific kind of substrate. So just like one key fits a specific lock, specific substrates fit specific enzymes. So they are highly specific. So this enzyme cannot catalyze a different molecule. So they are only targeting the molecules that fit them. So this is also why enzymes are named after the molecule that they target. So for example, if our targeted molecule was maltose, the enzyme that targets it would be called maltase. If it was lactose, the enzyme would be lactase. Do you see the pattern? Those both end with ASE. So most of the time, the suffix is replaced with ASE. The suffix from your targeted molecule is replaced with ASE, and that's the name of the enzyme that works with that substrate. Your active site, you also have to understand that the active site is the location that binds the substrate and catalyzes your reaction. Okay, so think of that word active. This is where all the activity is happening. So the shape of your active site is going to complement the shape of the substrate. Okay, so those are your key things you need to know about the shape of the enzyme, the components within an enzyme, and enzyme specificity. Another thing you have to remember, and we are going to repeat this throughout, is that enzymes remain unchanged and they're not used up. All right, what exactly are our enzymes doing? Okay, so if we look at this chart right here, along the x-axis we have reaction progress, and on the y we have energy. So before we can get our reactions started and take our reactants and convert them to products, we have to have a little bit of energy that will need to go in and kickstart that reaction. That energy is known as our activation energy. So what enzymes will do is they will lower that activation energy. So that kickstart that our reactants usually need before they can be converted into products is not going to be as big of a kickstart. So the enzymes have essentially lowered that activation energy for us, which will speed up the reaction. Now, it is important for y'all to understand things that enzymes are not going to do. These are common misconceptions. Enzymes are not changing the reaction. It stays the same. What would have happened, they're just helping it along. Enzymes will not make reactions happen that otherwise would not have happened. Okay, so they're not magic. They're not going to go in and make something happen that wouldn't have happened without them. They also will not change the energy of the starting point or the energy of our end point. The only energy that our enzymes are going to change is our activation energy. So why should we use enzymes? Okay, so in order to prevent imbalances of substances within our bodies, okay, so we need to make sure that all of the chemical reactions are being controlled. Okay, so the chemical reactions that are constantly happening within your body need to be controlled by your cells to make sure that nothing goes wrong. Okay, you never want to have too much or too less of any of your chemical reactions. So oftentimes this means that there is a need to speed up reactions. Now, we have two options if we want to speed up reactions. We can either increase the temperature or we can use enzymes. Okay, so our two options to speed reactions up when we need to are go up on the temperature or add an enzyme. Now let's look at increasing temperature. When we increase temperature, think back to what's happening to your molecules. All of the reactions are going to speed up because all molecules are speeding up in their movement. This means your wanted and your unwanted reactions. They're all speeding up. You can't control which ones. So that will lead to either damage to the cell or cell death. So this option isn't exactly the most efficient option. So our other option is to add an enzyme. When we add an enzyme, this is different than changing the temperature because we're targeting only the reaction that we want to speed up. And we're not messing with any of the other reactions. So think back to enzyme specificity. Okay, they're very specific, so when we're using them, we can make sure that we're only messing with the reactions that we really do want to speed up inside our cell. This can help us to avoid increasing the temperature and avoid the damages we would have gotten had we gone down the route of changing the temperature or increasing the temperature. So we're avoiding all of that harm. We're avoiding cell damage and cell death if we go down the route of adding an enzyme. So this is just a better option overall for your cells to control the chemical reactions that are happening. Induced fit. So researchers discovered that it's not a perfect fit. So when your enzymes are combining with your substrates, while all the models show this perfect fit where your substrate comes in and it locks in perfectly to your enzyme, it's not seamless. Your enzymes do have to slightly modify themselves. Okay, so they do slightly change their shape to accommodate the substrate that they're working with. Okay, so this is the term when we're talking about induced fit. Your substrates will induce your enzyme to slightly change their shape, and then it fits. Okay, so this fit is only possible because the enzyme is slightly changing their shape to accommodate that substrate. Now, since this fit between your enzyme and your substrate is so specific, and you know that your enzyme is already going in and doing that work to accommodate that shape and making that change, we need to remember that your enzymes will only operate under certain biological conditions. Okay, so it's a very strict guideline for how your enzymes are going to operate because of the fact that they need that very specific fit, and they need to be able to alter their shape to accommodate the enzyme that they are working with. So these biological conditions will come in a little bit. All right, enzyme substrate complexes. All right, your enzyme's job is going to be done by helping your substrates along. Okay, so they're helping to speed up the reaction by lowering the activation energy. So oftentimes this is done by the enzyme helping your substrates get into position and pretty much adding the pressure needed to help those substrates become the product that they need to be. So if you look on this picture, this could be more than one or more substrates. Now, using the active site, your enzyme is going to hold the substrates in place, and temporarily, this right here is what would be your enzyme substrate complex. All right, so in the process where your enzymes are helping speed the process along and speed up the reaction, that temporarily created compound is known as your enzyme substrate complex. Now, after the reaction is complete, your enzyme ends up releasing the product, and you're going to have your enzyme back ready to go, and it's not used up, it has not changed, it has not altered, it's ready to go in and do this process all over again because it's unaffected by the reaction. All right, so we need to know that your enzyme substrate complexes, these are the temporarily structure created when your enzyme is holding your substrates in place. Some of the other factors that are involved with our enzymes are going to be cofactors and coenzymes. All right, these are involved in activating enzymes and making them capable of speeding those reactions up. They're making them capable of actually binding to the substrates. So remember that it's very, very highly specific, so they sometimes do need the help. And these are called cofactors and coenzymes. Let's start with our cofactors. Your cofactors are going to be your inorganic molecules or your ions, and generally these are usually metal ions. So think of iron and magnesium. Those are your two most common examples. And also think about how these are used in bodies. You know how sometimes if you're anemic, you would take supplements with iron in them, or you do have magnesium. Some of the athletes might be familiar with magnesium because magnesium is often used with muscle cramps or like things that involve muscle contractions. Now, coenzyme are your organic molecules. So think of vitamins. Vitamins fall under your organic molecules. So this is why your vitamins, your daily dose of your vitamins is so essential, because they serve as coenzymes that are helping enzymes do their job. Now, regulation, as with everything else in your body, regulation is important even when it comes to enzymes. So your cells do need a way to control enzyme activity within the body. And your cells are going to do that by regulating the conditions in the body that enzymes are functioning in. Okay, so conditions that influence the shape of your enzymes are how your cells are controlling how much the enzymes are able to work. Okay, so remember that enzymes are very sensitive to that. If we mess the conditions in which they work, they're going to stop working. So your cells actually can do this in two ways. One, they can regulate the production of enzymes in the body, or the second way is that they can regulate the enzymes that have already been created. Okay, so enzymes that are already there within the body, they would need a way to regulate them as well. The first way, regulating the production of enzymes, your cells can do that by switching genes on and off that code for the production of those enzymes. So by switching it on, they can increase the production of the enzymes that are needed. Switching it off, cut off the production of enzymes that are already there and we don't need any more. Now, regulating the enzymes that are already made, this is a little bit more in depth that you need to know, and this is where your competitive and non-competitive inhibitors will come in. Okay, so we're going to use that inhibition to control the enzymes that are already existing. So first one is competitive. So in competitive inhibition, we're using the site that's known as our active site. Now, the other site we didn't look at before is the allosteric site within your enzyme. Now, in competitive, we're focused on the active site. In this type of inhibition, what's going to happen is we have a competing compound which will resemble your substrate. Okay, so this compound resembles the shape of the substrate molecule. So it has the ability to go in and compete with the substrates that are available in that environment. And when it goes in and it competes, it takes the spot that the substrate would have attached onto the enzyme. And if it's taking that spot, it's preventing a substrate from coming in and attaching to the spot. Therefore, it's lowering the activity of the enzyme because it's just there taking over the spot. So another substrate can't come in and the enzyme cannot help and a substrate produce or catalyze. Okay, so this is reducing the amount of product by preventing or limiting the substrates from binding to the active site. All right, so we can actually work around this. And the workaround to this would be the concentration of our substrates that are there. Okay, so when it comes to the competitive inhibition, we do need to know the concentration of our mimicked substrate and our actual substrate. Okay, so whichever we have a higher concentration of, we'll be better able to outcompete the other. Okay, so we have to know that that means that this is a reversible process and we can overcome the process by flooding the system with the substrate. Okay, so this is actually something that's commonly done in medicine, is you can either be given that mimicked substrate if you need external help with controlling the enzyme, or if your enzymes are, or if your body is automatically making too much of the compound that's resembling the substrate, the medicine can flood your system with the substrate, which helps that substrate better compete with your mimicked compound. Now, non-competitive inhibition, so think of that word non-competitive, so this one is not competing for the active site. This is also known as allosteric regulators. So in this one, we use this second site, which is our allosteric site. what happens is that we will have an inhibitor. The inhibitor will bond to the allosteric site. And when the inhibitor bonds to the allosteric site, what it does is it changes the shape of our active site. And when we change the shape of the active site, we're basically now completely taking away the ability of the substrate to bond with this enzyme because of that high specificity. Like, it's no longer going to work. So it's similar to if we were to change the lock. We've changed the lock, and now that substrate doesn't fit in that lock anymore. Okay, so in non-competitive inhibition, we are stopping our substrates from fitting in the spot by changing our active site. So there are two types to this. There are some that we can have activating stabilizers, or we can have inhibitors. So activating stabilizers would basically be when we need to alter our enzymes to allow them to work with certain substrates. We would end up adding those inhibitors, and it would transform the enzyme into the shape that's needed for it to actually work with certain substrates. Or the opposite, which is our inhibitor, that would be where we transform the shape so that it can't be used and we're locking away that enzyme. All right, factors that affect reaction rates. So we have two main factors y'all need to know. The first is temperature. Now, all enzymes have an ideal temperature that they're going to work best in. Okay, so there's an ideal temperature for all enzymes, and you can figure out that ideal temperature when you look at the graph. So you'll see graphs like this where you'll see the rate of reaction graphed against temperature. On those, at the very peak of your chart, that's the ideal temperature for that enzyme. Now, when we increase temperature for all of your enzymes, increasing your temperature will increase the rate of reaction to a point. Okay, so you see that point, like we got up to the peak of the graph, we increased the rate of reaction to a point. Now that point will be different for all your enzymes, but we are increasing to a point because essentially when we're increasing the temperature, think about what's happening with your molecules. We're increasing the chance of collisions by speeding up those molecules. So that's why to a point you do see an increase in the rate of reaction. However, if we were to change that temperature too much and add too much heat, we can end up damaging our enzymes. Okay, so right where you see where we got to that peak, when we go past that peak, that's now too much heat, and we have now started to damage our enzymes. And excessively high temperatures, generally over 42 degrees Celsius, will end up denaturing our enzymes. Okay, so that's our damaged enzymes that can no longer do their job because they have been denatured. Okay. When we say the word denatured, think of the D. Okay, denatured, basically we've changed its nature. Okay, we've changed its nature, so it's no longer doing what it needs to do. Generally, when we'll look at a visual, what you'll see is that it kind of falls apart. Your enzyme's active site will end up falling apart and changing, and that means that enzyme has now been inactivated. Okay, so the substrate no longer fits that active site, and so when the substrates can no longer fit those active sites, the enzymes can't really catalyze anything after that point because they don't fit our substrates anymore. Okay, so since they're not able to catalyze anymore, they're inactivated, they're no longer usable. Now, second factor that affects our reaction rate is pH. Okay, so same thing. Most all of our enzymes have a specific pH that they're going to function best at. And you'll see similar graphs like the one that we saw for our temperature, where you can use the peak to see the ideal pH for that enzyme. And if we have the enzyme at the wrong pH, what we'll start seeing is that the hydrogen bonds will be disrupted. And event that also does the same thing, where our enzyme structure becomes altered due to the hydrogen bonds being disrupted. So if the enzyme structures alter, again, it will not fit the substrates, meaning it has become denatured. Now, pH. Most enzymes are going to have the optimal pH at 7. However, that's not going to be all of your enzymes. It's important to remember that there are enzymes that will not have 7 as their ideal pH. Some enzymes will operate at lower pHs. So for example, pepsin, which is a digestive enzyme found in your stomach. Okay, so the enzyme pepsin will function at a pH of 2. And the same thing with lysosomes. They will also function best at a pH of 2. So think about the similarity between the two. Your pepsins are digestive enzymes in your stomach. Think about the pH in your stomach and then make that connection for your pH of 2. Same thing, what is the function of lysosomes? What are they doing in your body? Okay, and then they work at a pH of 2. So extreme pH leads to denatured enzymes. So I want us to think a little bit about this denatured. So we keep saying that enzymes have been denatured if we have extreme temperature and extreme pH. We're not using the word killed. Okay, that's important for you guys to remember, because enzymes essentially are not dead or alive. So since they are not dead or alive, they cannot die and they cannot be killed. Okay, since they're not living organisms. So they are biological molecules, are proteins made by living organisms. So when we refer to what's happening in the extreme pH and extreme temperature, that's why we're using our term denatured. We are inactivating them by changing their nature. We're not killing them. Okay, so that vocabulary I felt was something we need to kind of make sure that we are paying attention to. So this is it for 3.1, 3.2, and 3.3. Everything that y'all need to know about enzymes and factors that influence enzymes.